Greetings. I recently published 2 papers about the fission of membrane tubes. The short one, in Nature Communications, describes a cool new mechanism where fusion events can catalyze fission events. The longer one, in Biophysical Journal, is a more thorough description of the landscape of possible rearrangements for double-membrane tubes. As a result, I made a short video summarizing the former, and this is the longer video summarizing the longer paper. Links to both papers are, of course, in the description. Membranes are a defining feature of biological life. You're probably at least familiar with them as a barrier that acts kind of like a cell's skin and separates the interior and exterior. But compartmentalization is important for a lot more than that. If you look inside, you'll see a lot of organelles that act like cellular factories and waste disposal sites and much more. Each of these is enclosed in a membrane. Well, there are non-membrane bound organelles, and they're kind of a hot topic right now, but let's not focus on them at the moment. Biological membranes are composed of phospholipids like this. There's a head group that's hydrophilic and a hydrophobic tail group. When mixed with water, they spontaneously phase separate, forming structures where the hydrophobic tails separate and the hydrophilic heads go to the surface. This often takes the form of layers, but there are also a lot of other possible structures. I want to focus on these structures, mitochondria, which I think I legally have to refer to as the powerhouse of the cell at least once. In order to turn one mitochondrion into 2, you first grow it up big and strong, essentially duplicate everything inside, and then squeeze it in the middle. This results in 2 mitochondria connected by a tube. You then have to break this tube to get 2 individual mitochondria. If you ever tried to turn one balloon into 2 balloons by squeezing in the middle, you probably realize this is not easy. And just like your balloon responds poorly if you try to rip it and rearrange its connections, mitochondria aren't big fans of forming pores in their membranes either. To make matters worse, there isn't just one membrane, there are 2, an inner and outer membrane. So you're trying to fission a tube within a tube. This kind of double fission is the focus of this video. Normally, the fission of these tubes is catalyzed by proteins that wrap around the tube and squeeze them. I won't go into the protein involvement in this video, but to a first approximation, you can think of it as simply reducing the radius of the tube. I can get the same effect from applying a tension, so I'll use tension-induced constriction as a proxy for protein-induced constriction. There are many ways that I can simulate membranes, and thus the process of double-membrane fission, and if you've watched other videos on this channel, you're likely familiar with some. In that case, you'd probably prefer to skip ahead to the results, but to summarize, at one extreme there's molecular dynamic simulations. These represent each particle in the system, and the membrane emerges from the statistics of the lipids. These calculations are very computationally intensive, so it's difficult to capture slow processes in large systems. At the other extreme, there are Helfrich models. These ignore the internal degrees of freedom of the membrane and treat it like an elastic sheet that can deform in various ways. The energy of the sheet is written in terms of the degree of each kind of deformation, and you can study the behavior of the sheet. The issue with this is the ad hoc nature of the energy functional, and the difficulty is studying changes in topology. The method used in this talk lies somewhere in between, where the membrane is emergent from lipid statistics, but instead of getting those statistics from the behavior of individual particles, they're calculated using a statistical field theory derived for polymers. A more thorough description is given in my other videos, but in short, we can write the partition function for a set of particles composing our membrane as the sum over Boltzmann-weighted states. The energy in the Boltzmann factor comes from the bonded and non-bonded interactions between particles. The way I've written it may look unnecessarily complicated, but see the video about polymer field theories for the reasoning. Without getting into any of the mathematical details, and also glossing over some important parts, I can write the partition function as a sum over configurations of the system, where a configuration is defined as some function phi of r, which gives the concentration of lipids at every position in the system. All of the internal degrees of freedom and the multiplicity of states that can create the concentration profile comes in through this effective Hamiltonian. If I want to study a particular state, that is, a particular membrane geometry given by a concentration profile, I can stick a functional delta function in this partition function, pulling out a particular configuration, and we see that the free energy of this configuration is given by the effective Hamiltonian. In practice, what I do is start with a profile given by this phi. Really, I should have written a set of phis giving the concentration of each molecular species, hence the gamma subscript. I then calculate a potential field, W, which is felt by the test molecules in this configuration. I can then calculate a probability distribution over polymer configurations. I've used P as a shorthand for just calculating probabilities in polymer statistics, but this notation is a bit ad hoc. In short, I can choose a monomer labeled S and fix it at spatial position R, and calculate the partition function for the portion of the molecule on either side of S. If I multiply these 2 partition functions together, that gives me the Boltzmann-weighted number of ways to place monomer S at spatial position R, and is proportional to the probability of placing a monomer at that position. If many molecules are sampling this distribution, then this probability is proportional to the average monomer concentration. If I then integrate this overall space, I get the partition function for the entire molecule, and I can combine it with the product of the partial partition functions to get a unitless concentration. The concentration of each molecular species is then given by a sum over the monomers that compose that species. So I started with a system configuration, and I calculated what configuration the system wants to be in. If they're the same, then the system's happy. It's in a stable or metastable configuration. If they're different, the difference gives me local exchange chemical potentials that then drive the evolution of the system. I don't usually say this when giving these talks, but this is a bit of a simplification. In practice, I actually use the chemical potential fields as the primary thing being evolved. They're conjugate to the concentrations, so you can think of it as equivalent to evolving the concentrations, but it's an important technical subtlety. If I iterate this process starting from some configuration, I can get all of the expected equilibrium states of membranes. Planar membranes, membrane tubes, vesicles, micelles, worm-like micelles, all of these things. But I'm not just interested in stable states. I want to know how one state transforms into another. If the system's simple, I can use a simple reaction coordinate. For example, I can write the free energy of a pore in terms of its radius, and it has this general shape. I can go on to find an analytical expression for the free energy as a function of the radius. I can map this onto SCFT if I want, or I can find the macroscopic properties, things like the line tension and membrane tension, and I can describe this easily using that one simple reaction coordinate, the radius. But for the fission of a tube, it's not clear a priori how this reaction even proceeds, far less what nice reaction coordinate I can use. So I turn to a more general method, the string method. I can think of the concentration at each point as a variable used to describe the system, or equivalently as an axis in a configuration space. The state of the system is then given by a point in this space, and the transition pathway is given by a path through this configuration space. In practice, I represent the system with millions of points, but I can't draw millions of dimensions, so here's a cartoon with just two. There are free energy minima representing stable states, as well as high free energy states. An equilibrium SCFT calculation is done by starting with one state and evolving it such that it relaxes into a close-by minimum. This can be viewed kind of like a gradient descent method. But if I want to describe the transition from one state to another, I need a set of points, creating a curve through this configuration space. I start off with some guess for the pathway. In practice, I come up with something that kind of makes sense, and create a bunch of concentration profiles that follow that possibly sensible pathway. I then relax it under the constraint that each point remains equidistant in configuration space. This has to be done carefully, but if it's done correctly, it produces the minimum free energy path through configuration space. Note that this path is only locally optimal, not guaranteed to be globally optimal. So I need to try a bunch of paths and kind of mess around with it a bit to try to be sure that the path that I create is actually a good one. Here's an example of an optimal path between one large vesicle and two smaller ones, i.e. the pathway for vesicle fission. And here's the fusion of a vesicle with a membrane. Note that these paths are fully reversible, so it's also the optimal path for vesicle budding. Relevant to some other work is what happens if the vesicle stops and reverses. See my work on the kiss and run mechanism for neurotransmitter release for details. One important thing that I want you to notice is that each of these processes culminates in the formation and fission of a neck connecting different compartments. This is a very common feature in topological transitions in membranes and a key step in recompartmentalization. So let's look a bit more closely at the membrane tubes. We can start from a membrane that's under tension. The tension is controlled by the lipid chemical potential. The tension also controls the radius of the tube. The higher the tension, the smaller the tube. I can calculate the optimal path for fission, and it looks like this. The tube first constricts locally, then forms this connection blocking the tube. The connection relaxes. It then may elongate into a worm-like micelle, or it may not. It tends to elongate like this at high tension, but doesn't at low tension. This calculation was done at high tension. The connection points between the worm-like micelle and the tube are thinner than the equilibrium thickness of the bare worm-like micelle. They're therefore easier to contract and break. So this one contracts here. The worm-like micelle thins and breaks at one of these connection points. It then retracts, and being under tension, the tube also retracts. I'll show this one more time because I like the video. In addition to the pathway, I also get the free energy for every point along the path. I can calculate these directly from SCFT or using the chemical potential along the direction parallel to the path. Comparing these two ways kind of gives me a sanity check for the free energy of the pathway. The horizontal axis is the distance along the path in terms of an abstract reaction coordinate. For more details, see my video on the string method. The curve starts off with a free energy of 0, which I define as the free energy of the unperturbed tube, and it increases to get to the peak. This is where the connection forms. The connection then relaxes, the worm-like micelle elongates, then it breaks off here. This is where the worm-like micelle retracts, and here is where the capped-off tube retracts. In addition to these calculations, we can also do experiments to look at the tubes during fission. And by we, I mean my collaborators in Spain, Anna Shanova and her group. I'll give you more details shortly, but in short, they put tubes under tension and let them undergo fission, freezing them at various points throughout the process, and thereby getting snapshots of the fission while it's happening. When they do that, they see all of the key structures that I find in my calculations, which is pretty cool. I can repeat these calculations at different tensions. At lower tension, the free energy barrier, given by this height, is higher, which makes sense. A wider tube under lower tension is less likely to spontaneously break. Also notice that the worm-like micelle portion is barely there. I mentioned that the worm-like micelle may form, and whether it does depends on the tension. At low tension, the worm-like micelle has a higher free energy than the tube, so the system can't lower its free energy by converting the tube into worm-like micelle, and the stalk breaks off before it can extend. At higher tension, the barrier is much lower. This negative slope occurs where the worm-like micelle grows, because the system can lower its free energy through the conversion process. One thing that I want to point out here is that the order parameter from the string alpha reflects changes in the local concentration and does not directly relate to any intuitive order parameter. During the first increase, the change comes from local changes in the tube radius during constriction, but that doesn't vary linearly with alpha. There's a more thorough description of intuitive order parameters in the paper if you're interested, and I encourage you to check it out. But the most interesting part is that if I map the free energy change onto the local radius change after a very brief quadratic dependence, it's linear. That is, the free energy varies linearly with the local constriction for all tensions, and with approximately the same slope. This suggests that the force needed to create the local constriction is constant and independent of the radius. One other thing that I want to mention, because it comes up almost every time I give a talk about this, is the possibility of poration in the fission mechanism. It has been seen in particle simulations, and it's possible. I've done calculations that explore this possibility, and they're discussed in the paper, though not in this talk. The short version is that it looks like poration is unfavorable because of the high edge tension of the pore, but it's possible, especially if there's something opening a pore from the beginning. Since there is a dependence of the tendency towards poration on the membrane edge tension, as well as the bending and Gaussian curvature modulus of the membrane, which are discussed in the paper, and these depend on lipid architecture as well as the environment, it may simply look unfavorable in this particular model and parameter set. But it does look very unfavorable. And this model and these parameters show general behavior that I expect in other circumstances, so I'm skeptical of the pore pathway. Okay, so I've told you all about the fission of single tubes, and validated the method at least qualitatively using experiments. So now it's time to turn our attention to double tubes. Double tubes are more complicated because, well, there are two of them, and they can do things that single tubes can't. I'll get more into detail on that later, but first let's look at the simpler things that it can do. I can, of course, do the calculation for the straightforward fission of the inner tube via the previous mechanism. I used a pretty high tension for this calculation for reasons that may become clear shortly. Also note that the graph doesn't quite look the same as before. The worm-like micelle growth portion is more curved. Since the outer tube also changes, measuring the L2 norm for changes along the single tube pathway does not lead to the same reaction coordinate as the double tube pathway. Alpha can be improved upon by using a reaction coordinate like the radius of the tube at its most constricted point. I haven't shown the results in this video, but there's a discussion in the paper. In short, it gives a better and more physically intuitive reaction coordinate. The interesting thing about double tubes is the additional degrees of freedom. They can do things that single tubes can't. An obvious example is the fusion of tubes. A hydrophobic bridge can form between the two tubes. From this graph, it doesn't look like the stalk is metastable since the free energy increases monotonically, and it doesn't form a minimum once it's formed, but we have reason to believe it's actually metastable. If we repeat these calculations in the canonical ensemble, where the diffusion of lipids is effectively restricted, then the stalk becomes metastable. It's only very weakly metastable, as you might expect from this change on ensemble changing its stability, but it's metastable. As you'll see later, it's also metastable in particle simulations. Once the stalk forms, it can expand, forming a hemifusion diaphragm. Once again, it looks like a free energy plateau, but we think that it should be metastable. Once again, this is because when we run the calculation in the canonical ensemble, it looks metastable. And again, in the simulations that I'll show you later, the stalk and the diaphragm are metastable. The additional freedom allowed by this Model A grand canonical ensemble calculation simply allows too much freedom for the lipids. This is only a small perturbation on the real behavior, but for very weakly metastable states like this, it ends up mattering. The system also has other options starting from the stalk state. Rather than expanding the stalk into a hemifusion diaphragm, it can use it to catalyze the fission of the inner tube. This fission-by-fusion mechanism seems like it might help explain some results seen by experimentalists with whom we've been collaborating, and it's more relevant at lower tensions than the ones shown here. For details, check out the other video on double tube fission, or our paper on the topic, both of which are linked in the description. The hemifusion diaphragm can also act as a jumping off point for inner tube fission. This pathway has a higher free energy barrier since it involves formation of the hemifusion diaphragm, and it's also only available at high tensions since it involves the formation of an elongated worm-like micelle. Nonetheless, I think it's pretty cool. Once the hemifusion diaphragm is formed, it can also continue to grow around the tube. The free energy barrier is quite large, and given that there are other available avenues with much lower free energy barriers, I don't expect this pathway to be taken often, unless something intervenes to cause it to be more favorable. Biology does weird stuff sometimes, so I wouldn't be surprised if it happened under some circumstances. The end result is what I've been calling a circularly hemifused state. Once this circularly hemifused state forms, it has a few options. Given that the tubes are under tension, the system can lower its free energy by simply fully elongating the single tube region, that is, pulling the junctions apart. But it can also break in a number of ways. The option with the lowest barrier, as far as I can tell, is to disconnect the inner tube. As usual, there are several ways this can happen. One of them involves forming a pore at one of the fusion sites and unzipping the connection. Since the stalk is metastable, the inner tube can remain connected for a while, but eventually it disconnects. The paper discusses other fission mechanisms, including the poration of the single tube and collapse of the inner tube, i.e. the previously discussed mechanism for the fission of a single tube. Putting this together, there's a large landscape of possible rearrangements. I've simplified it by combining different pathways and ignoring complications. These are discussed more in detail in the paper. Essentially, we can separate the set of possibilities into a set of metastable nodes and connections between the nodes. That is, represent the landscape of possibilities as this graph. Each of these sub-pathways may or may not be reversible and has a barrier corresponding to the minimum free energy through the configuration space between these local free energy minima. In case I haven't made it clear, this work is about understanding these metastable states and finding pathways between them, including the free energy barriers, in order to understand how the system can evolve step by step, and also understand how biology may change these barriers in order to change the pathways, and therefore final states. A path through configuration space corresponds to hopping from one node to another. For example, this path gives one possible fission mechanism. It should also be noted that a lot of these paths are reversible, so can be interpreted as forwards or backwards. I've alluded to it a couple of times, but there are some limitations to the use of SCFT in the string method. SCFT is a mean field theory and ignores fluctuations. The Gaussian chain model also simplifies the behavior of lipids. My implementation of the string method uses Model A dynamics, which may not accurately reflect the way that lipids actually move around. This is not to dismiss my work, but to acknowledge its limitations, as one should with any approach to any problem. Fortunately, I can test these approximations. Using a simulation software Zoma developed in the Muller group, I can run particle-based simulations that can be effectively mapped to the SCFT calculations. They include fluctuations since there are a finite number of interacting chains and the chains diffuse around, so follow a more realistic dynamical model. I initialize it by starting the molecules at random positions and then imposing the SCFT field on them to force them to assemble into the geometry that I want. I've glossed over a bit of the necessary fiddling around, like how I get the lipids to come in from far away, but these are the broad strokes. Once initialized, I can turn off the fields and simply let the system evolve. The top video shows the Zoma result, and the bottom shows the SCFT with string method result. Since there's no explicit time in the string method, and there's no activation time, I adjusted the speed and inserted pauses to line them up. Nonetheless, it seems that the Zoma simulation follows the path predicted by SCFT. I've done a lot more of these comparisons. Once again, see the paper for details. In this work, I used SCFT to model changes in geometry and topology of membrane tubes, particularly double membrane tubes. I showed you a simple leakless pathway for fission of a single tube, and went on to explore a landscape of possibilities available to double tubes. The presence of a second tube opens up a lot of new possibilities. The most notable is the fission-by-fusion pathway, but that's described in more detail in another video. The point of this video is to show you the plethora of other possibilities and pathways, and to get you to think about this landscape of possibilities as a graph with metastable nodes connected by transformation pathways. I also want to point out this state, which I call the circularly hemifused state. I wouldn't be surprised if it was described elsewhere, but I don't recall seeing it, and I think it's pretty neat.